Hello, good morning, everyone. Can you
Just a
see
second,
my...
I haven't,
Okay.
Yeah, no, not yet. I haven't, I need to move this, sorry.
No problem.
Okay, I'm also turning you so that you see the audience.
Wonderful. That's great. Thank you.
And I'm trying to, yeah, just a second, still not full screen. Okay, now we are full screen. Now everyone sees you. Hopefully you see everyone.
Yeah.
All good?
Nice
Okay.
room. Okay. Well, good morning, everyone. I'm Jessica Zhang from Carnegie Mellon University. Before I start, I would like to thank the organizers to invite me to give this talk. And also, I apologize for not being able to be there in person. So then hopefully next time we can meet, somewhere in person. Today's talk is mainly about our latest project on PDE-Constrained optimization and machine learning enhanced simulation for material transport control in neurons. So this is collaborating with my PhD student, Ran Li. So neuron morphology is very complicated. It looks like, the tree structures. A typical neuron structure consists of a cell body with a lot of dendrites, those small branches, and with a long, slim, long axon. At the tip of the axon, you can see, a lot of terminal bulbs or synapses. And in this way, the neuron exhibits a highly polarized structure. And it's important to know the material synthesis and degradation mainly happen in the cell body. And in this, in this particular, situation in our neuron system, the intracellular material transport is very important. So it needs to transport the organelles from one location to the other at the timely manner to support a neuron's survival and function. Sometimes the disruption may happen. Such disruption will lead to the neurological and neuron degeneration disease, for example, Alzheimer's disease, Huntington's disease. So that gives us, the problem, the motivation, it's an important problem, we wanted to study. So from literature, we found this video, from the paper in Neuron 2011. If you look closely, in the video, you can see some particles are moving along the neuron. They see the bifurcation structure here. So this organelle is called mitochondria. So that is mainly to generate energy for cells living. There are many different types of the organelles in the neuron. They need to be transported along the neuron, structures. So the neuron morphology is so complicated. So which means that the organelles need to be smart enough to transport, especially when they meet the bifurcation joint location. So the organelles need to, take the right path and to go to the right place. And how do we study this? How do we simulate material transport? And that is the key issue, in our research project. So this problem has been studied in some literature, but people only use the 1D model. And there are two kinds of transport in this system. One is passive diffusion. For example, in the cell body and also synapse, sometimes also along the axon, the organelles or materials, they are free to move around, through the diffusion process. Another type of the transport that is along the neuron axon. So the axon, you can see a lot of those green lines, they call microtubules. So the organelles can attach to those microtubules, and then just like we are taking an escalator, so we can move fast along the microtubule. So that kind of transport is called active motor-driven transport along the microtubule. So you can see, those two types of the transport forms, all the transport phenomena in our neuron morphology. So people use the 1D dynamic reaction convection problem and numerically solved it using the finite difference method. So they modeled 5 states. So U0 represents the passive diffusion, which means the materials don't bind to the microtubule. But sometimes along the axon, so U1, U2 represent the organelles attached to the microtubules, but one is in the pulsing state, one is in the moving state. U-1, U-2 are in the opposite directions. So when we have the initial condition, by solving this 1D dynamic reaction convection problem, we can simulate, compute after 2 weeks what the concentration profile looks like. So in our earlier study, we extended this 1D model to 3D domain. And we solved this reaction convection diffusion problem. But in addition to this, we also model the velocity here, V plus minus, in a more accurate way. In the 1D model, people just assume a constant velocity in their study. But here we build another Navier-Stokes equation solver to compute the velocity and couple it with the reaction convection diffusion. Then we can accurately include the velocity information into the transport. So in our solver implementation to enable stability and convergence, so we implemented a variational multi-scale method and also SUPG method, so that can give us stable results in the numerical solver. We use a high-order finite element method. It's called the isogeometric analysis. So that is the connection with the geometry. So we use the spline basis functions. So in the model here, from the neuron morpho database, which is a public domain database, so we can download the central line information or skeleton information with the diameters. And we have the sweeping method, skeleton-based sweeping method. We can build the single pipe and the bifurcation. So we define the templates to handle the bifurcation structure. And then we can generate a hexahedral control mesh. And using the hexahedral control mesh as the input, we can build the truncated hierarchical B-splines, so which is the spline basis function we use for our isogeometric analysis solver, which means that is the finite element basis function. And in the parameters, so we use those parameters from experiments, we got from the literature, and we include all those parameters into our IGA solver. So we have a Navier-Stokes equation solver and also the convection diffusion, reaction solver. And couple them together, then we can compute the velocity and the concentration in the complex neuron tree. For the spline models, since this is a geometric modeling deep learning workshop, I wanted to explain a little bit what kind of spline basis functions we are using. So we are using the truncated hierarchical B-spline. This idea, is based on truncation mechanism. So suppose at level R, we have those basis functions, and at N4R, you will want to refine it locally. We replace it with 5. Suppose we use a cubic spline, so we replace it with 5 children basis functions. So, but when we replace that, we will have a duplicated contributions from the some children basis functions in the between those 2 levels. And that gives us the problem of the partition unity. So which means if you directly replace N4R with 5 children basis functions, we lose the property of partition unity. So then to resolve this issue, you can truncate the neighboring basis functions, N5R, N6R, the same as N3R, N2R. After you truncate it, truncation means you remove the children basis function with duplicated contributions. So if you implement this, you can, continue, you can keep the property of the partition unity. So this work is restricted to rectangular parametric domain, and it cannot handle extraordinary nodes. So in our study, our geometry is very complicated. So our domain is arbitrary. So we need to handle extraordinary nodes and also to support the local refinement. So firstly, we extended this idea to T-splines. So what we did that is we compare one regular, structure, and we want to locally refine these two blue elements. So to enforce the nestedness property of the spline spaces, so we extended these two edges. So we got those two red edges, and then we can preserve the topological constraints and to ensure the nestedness property. So after that, we check which basis functions are partially refined, and which means that is between two different resolutions. So they need to be truncated. So all the blue ones, they are in the final level, we don't need to truncate, but all the red ones, we need to truncate the basis functions in order to preserve the partition unity property. So then we need to detect which children basis function has a duplicated contribution, and we remove that from those to be truncated, basis functions. So how to detect that, the idea is straightforward. So we compare the given control mesh before local refinement and also the control mesh after local refinement. And if that children basis functions is defined, very well defined on the refined control mesh, then we just remove it from the original basis function. So you implement this idea, then basically you can apply the truncation mechanism on T-splines. So using this idea and together with the extraordinary nodes, we can handle complex geometry. So the extraordinary nodes, we know it has, other than 4 elements sharing one node in 2D, which means the parametric space, the quadrilateral tensor product. So for this one, we couple the node interval duplication and keeping the Bezier patches, with some optimized coefficients, we can enforce that. And, in 2D, you have the clear definition of the normal direction. So we can also get G1 continuity. But for this study, we don't allow extraordinary nodes to be within the two-run neighborhood, but it can be on the boundary of the two-run. So we also use the uniform node intervals around the extraordinary nodes. So this idea has been extended to 3D. For 3D, since the inside the volume, the normal direction is not very well defined, so we just keep C0 continuity for the 3D extraordinary node. But we can get a convergent result, and it is analysis suitable for the isogeometric analysis. So for this idea, there is one drawback, and even it can give you the convergent result, but the speed of the convergence cannot reach the optimal convergence rate. So, in the next step, we started a blended B-spline construction study. So we added some basis functions in the surrounding local area around the extraordinary node. And then what we did that is in 2D, we add C1 functions, basis functions at the face points. And also we add C0 functions at the extraordinary node and also edge point, those blue triangle on the spoke edges. And in 3D, similarly, we add C1 continued functions on the body points. And then on the spoke faces, edges, and the extraordinary nodes, we add C0 functions. By adding those active C1 C0 functions into the construction in that local area, we blend all those basis functions together. So we already proved that we can get the optimal convergence rate for both quadrilateral elements and hexahedral elements. So that is the basis for our neuron computation. So we have actual nodes, we have truncated hierarchical B-splines that can support our study to solve the convection diffusion reaction problem coupled with the Navier-Stokes equation. So we tested our solvers on the many complex neuron trees, and here are 3 examples. So we can compute the velocity, we can compute the concentration. You can see the local features of the flow pattern if you zoom in and check details if you want to study a particular area. So in our neuron morphology, we know not always be the normal transport, and people, in researchers in experiments, they have already found some, abnormal transport issues. For example, in this study, so people find microtubule swirls, and that form this kind of swelling geometry and complex swirling, the flow pattern. It was mainly introduced by a particular protein. It's called the tau protein, and it introduced impairment of organelle transport. So this is mainly because of the polar reorientation of the microtubules along the axon, or their displacement to some memory domains. So this introduced traffic jams, which reflects the accumulation of organelles, and those accumulations are generally observed at the point of the microtubule polar discontinuation or mismatching. So that is the phenomena people observed, in the experiments. In another study of the Huntington disease, the similar phenomena was observed. So you can see those white regions and those are the aggregates. So that was introduced by a particular Huntington protein. So those kind of Huntington protein forms insoluble aggregates. So they can accumulate in the soma and also axon. So such, accumulation or aggregates can physically block transport of organelles along the axon and then bring us trouble. So that is, the main issue we want to simulate. Can we simulate the traffic jams and the swirls? This, we found some evidence from the experiments. So in our research, we developed this new model. It's called the PDE-Constrained optimization model to to simulate the traffic regulation from neurons, and then we can control the transport process. In the objective function, there are 3 terms on the right-hand side. So the first term you can see is defined based on the velocity. Capital V plus minus is the desired velocity. So given certain input, settings, so initial condition, boundary condition, and all the parametric, parameter settings, we can use the Navier-Stokes equation, to calculate this desired velocity. The second term is to control how much we want to optimize the transport to avoid the traffic jam. It's defined based on the derivative of the concentration. And we have a parameter alpha. You can, user can give that how much you want to optimize. The last term is the regulation term, is defined based on the control force, F plus minus. We also give a parameter beta here. So in the constraints, we have 3 PDEs. The first PDEs are defined based on the previous motor-assisted system. So that's the reaction, convection, diffusion problem. And the last one is to model porosity. And then we use all those 3 together. And when we solve the problem, the PDE-constrained optimization numerically, we implemented the all-at-once method and build the KKT conditions. And numerically, we use the GM-RES solver in PET-C in the implementation. So the traffic jams can be modeled by introduced into the system by setting certain parameters. So there are 2 kinds of traffic jams. The first category is related to reduction of motors. we can modify the attachment rate and the detachment rate to model this kind of traffic jam. The second category is introduced by abnormal microtubules. So for this type of the traffic jam, we can modify the distribution of R plus and R minus. So for example, there are two kinds of, this kind of traffic jam we want to study. One is a material trap, one is a microtubule swirl. We give R plus, a drop in the local area in the R plus and a jump in the R minus. Then we can model the material, trap. Or we can model the reduced number of microtubules, how this can introduce the traffic jam. So another one that is a microtubule swirl. So you see this swirl pattern is very complex. So we need to form this kind of, swirl pattern. And it also comes with swelling geometry. So we can model the swelling geometry easily. And we use R plus, R minus, this kind of profile to model, to introduce this kind of a swirl pattern. So using those ideas, so we are more interested in the microtubules related traffic jam in this study. So we focus on the second category, but we model both material trap and the microtubule swirl. And for those, transport, we have three conditions. One is a normal transport, one is a traffic jam caused by reduced number of microtubules, all the material trap. The third one is the traffic jam caused by the microtubule swirls. So in the normal transport, to make the problem easier, simpler, we assume a unidirectional transport with a unipolar microtubule system. So which means we only set R plus, we allow the material to transport from a cell body to the synapse. But the system is, it supports a two-direction transport. You can include both of them. So for the first traffic jam caused by the reduced microtubules, we give a sudden jump in R plus. So you can see here in the middle region, and to model this kind of traffic jam. For the microtubule swirls, it's very complex. What we did that we look at one cross section in the swelling geometry. And on the cross section, we select the red regions and the blue regions. So red region, blue region, we define different configuration for the R plus, R minus. And in order to make sure they form a swirling loop in that local area. So by using those ideas, then we can model the microtubule swirl. So then the following, we tested those ideas in the single pipe. For this pipe, and to introduce the traffic jam from the reduced microtubules, we give R plus a drop here. And then comparing with the normal transport velocity, you can see here there is a blocking. The velocity slowed down in the magnitude, but the direction doesn't change. If you see the the 3D arrow directions. In the concentration, we also plot the concentration along the center line. The normal transport, you see there is no significant change along the center line. But with a traffic jam, you see aggregation, accumulation of the materials here, the red region. And in the concentration plot, you see a jump. And the same phenomena you can observe in the 3D. So you see the accumulation, the red region here, and we have the concentration jump here. So in our objective function and constraints, we have several parameters. So here we study how those different parameters influence the transport. So the alpha and the beta, they control how much we want to optimize to avoid the traffic jam, and the beta is to regulate the system. So we found that when we have a smaller alpha value and smaller beta value, we get a more serious traffic jam. That also makes sense. So which means you optimize less or you regulate less. So in addition to that, we also studied ratio K by K prime. When we increase this ratio, this makes more material attached on microtubules, but slowed down due to the reduced number of microtubules. So on the other hand, when we decrease this ratio, it can make more material transported, through the slow diffusion. So this kind of a phenomena can also be observed from the velocity profile. We also tested this idea in the tree structures. So we take a small portion of the tree, this part, and we introduce the traffic jam by the reduced microtubing in this local area. And then we compare the velocity with the normal transport. You can see there are some blocking here. And in addition to this blocking, you can see when this local area is blocked, and more particles move along the other direction, try to avoid the traffic. It's just like you drive a car on the road and you found some, traffic problem, ahead somewhere, you take another direction. And in the concentration, we plotted from the inlet to each outlet, and we can plot the concentration distribution along the center line. What we found that if we have the, the branches that's downstream of the blocked area, so like pipe 1, 2, 3, 4, those are downstreams. And for those, downstreams, we found a jump in the concentration because we have a traffic jam in this area. And on the other hand, in the pipe 5, since it doesn't go through the blocked area, we don't observe, a serious, obvious jump here, comparing with the, normal transport. So this shows another, similar study, but a different geometry, more branches. So we introduce a traffic jam here, and for the downstream, for all the branches 3, 4, 5, 6, 7, 8, you observe the jump. But the other directions which are not downstreams, you don't observe obvious jump. But you see some difference. This is because traffic jam happen here. More particles, more materials choose to to go through the other pipes. So the next, we want to study the traffic jam introduced by the microtubule swirl. This is more, the flow pattern is very complex. To model this, we modify R plus, R minus. And in order to form this counterclockwise, swirl pattern on the cross section, R plus, R minus, we defined to be non-symmetric. So in order to have a counterclockwise, pattern, if you want to model clockwise, then you just swap, the R plus, R minus. So in the velocity, we compare with the normal transport, you can observe some swirling, pattern here, and the same thing you can observe in the concentration profile. To see more closely, we plot the velocity streamlines. So the normal transport, you can see those beautiful streamlines. And in the microtubule swirl introduced traffic jam, you see those vortex patterns and some swirling, flow over there. 3D, we use those red, blue, definition on the cross section. And in the microtubule swirl, you see the normal one is like this, regular transport, but microtubule swirl, you see, those, irregular flow and the vortex pattern. So it's interesting to see that those vortex pattern generally happen in the high concentration region. That is an interesting observation we get from the, numerical results. For the microtubule swirl, since it's very complex, pattern in the 3D, we designed 4 different configurations on cross section. You can lay out different regions for red and for blue, and then you can study what the swirl pattern look like for that and compare with the normal transport. For each red region, blue region, we can also extract the center line and then to show the concentration distribution. And we can see at the blocked, swirling local area, we have a jump. And we observe the reversing and the vortex pattern streamlines. Those are caused by the distribution of a different direction of microtubules. We also found that the reversing streamline mainly occurs between the red and the blue regions. So this indicates the transport paths of the material is extended because you need to move along direction or even trapped in the local area. So more studies on the complex tree structure with the swelling geometry and the microtubule swirl, and from the vortex, the streamline, you can compare with the normal transport. You see those vortex pattern, and this shows another structure. So we observe a vortex pattern. They happen at the high concentration region. That is interesting. For this tree study, we have the normal transport, and we introduce the traffic jam from the reduced microtubules, but no swelling geometry. And the last study is with swelling geometry and microtubule swirl. So you can see the flow pattern, the directions, look like in a streamline. And in the concentration profile along the center line, we also observe when it is downstream of the blocking area, we observe the obvious jump. But in the other, branches, we don't observe this. The same thing you can see here. Number 6, number 7, we have a jump, but here we don't. So the isogeometric analysis is a high-order finite element method. So we use the cubic spline basis functions. But this method, we need to build a linear system, and then we solve the large linear system, AX equal to B or KX equal to F. And we have the time iteration, so that gives us a very expensive solver. And for the deep learning workflow, you can, collect the training data. You can use a neural network to train, and then using the trained data, you can quickly give the prediction. So then, which means, if you can build a surrogate model and to quickly give the prediction, that will really, provide a timely, quick, fast, prediction for the transport phenomena, and also concentration, provide those useful information to the physicians. So that is the motivation why, we use deep learning to study this together with our isogeometric analysis solver. So we built this IGA-based physics-informed graph neural network in our study. Since our neuron tree structure is unstructured, so our mesh is complex, we use spline basis functions, so we cannot use CNN. So, but the good thing that is the tree structure, it only consists of pipes and bifurcations. So we decompose the tree into 2 simulators, pipe simulator, bifurcation simulator. And in the pipe simulator, we assume, we have a traffic jams, but in the bifurcation, only normal transport. So in the pipe simulators, we allow to have a normal transport, traffic jam caused by reduced microtubules, traffic jam caused by the microtubule swirls. Bifurcation, only normal transport. So we also have a sampling model to assemble all the simulators together to form the whole tree. So this is basically our structure architecture for our IGA-based, physics-informed graph neural network. So in our simulators here, we use a GNN block and a multi-layer perceptual decoder scheme. And since we have a truncated hierarchical B-splines, we use the Bezier extraction. And those Bezier extraction matrix information are very useful to compute the loss functions. So in order to learn underlying physics, we include the residuals from our equations into the loss function. Our loss function includes 3 components. One is measured from the prediction at the sampling points. We also include the residual from PDE constraints. So the third one is we evaluate the objective function from our system. So in the following, we also use the recurrence method, to give the, prediction, improve the accuracy. For the assembly model, we assemble the predicted result from the simulators. To keep the interface between two simulators consistent, we introduce the penalty term into the loss function. This is the second term here. So, we have only single pipe and bifurcations in our neuron tree. So in the assembly model, we have 3 types of this assembly: pipe assembly, pipe bifurcation assembly, and also bifurcation assembly, because we observe sometimes the 2 bifurcations are very close to each other. So we allow bifurcation assembly. So with this architecture, so we build the training model. So we collect all the training data. So we extract the simulation parameters, velocity, initial boundary conditions as the input. The output includes the velocity and also concentration at each node. And we have 3 pipe simulators, and that includes a normal transport, 2 traffic jams, and we also have a bifurcation simulator. Each simulator, we created 1 million samples. So consisting of 100 geometries, 100 boundary conditions, and 100 sets of the alpha beta values. In the assembly model, each type, we have a pipe assembly, pipe bifurcation assembly, bifurcation assembly. Each type has 300,000 samples. So with 30 geometries, 100 boundary conditions, and 100 sets of alpha beta values. So then following the standard way, we conducted 4-fold, deep learning validation, cross validation. And then we got for the pipe simulators, we got 94%, 93, 92% accuracy. Bifurcation simulators, we received 94% accuracy. Assembly models, we have a 91.7%. So with all those trained, optimize the parameters, giving a new train, new settings, boundary conditions, we don't need to rerun IDA solver, and we can use those deep learning methods to quickly give us the prediction. We can also compare with the ground truth, which we use IGA simulation result as the ground truth. So we tested, a number of, complex tree structures for the normal transport. We all got 90% above accuracy, and the speed, we can speed up 120 or even 300 times faster. So this shows some comparison for the normal transport. We also tested on the microtubule, introduced traffic jams. So those are 2 types of the traffic jams. We started using the, the neuron network graph or neuron network, and we got 93%, 92%, 90%, 91% accuracy for both, traffic jams. So with, the introduced by the reduced number of microtubules, or introduced by the microtubule swirls. So in conclusion, and we built this IDA-based PDE-Constrained optimization, and also we used physics-informed graph neural network to give the quick prediction together with our IDA solver. And we observed that both geometry or morphology of the neuron and microtubules are very important, to influence the material transport. So the reduction of the microtubules in a local area can slow down the transport velocity and lead to traffic jam in that particular area. The swirl of microtubules in a local area that together with the swelling geometry can extend the transport distance and also trap the material in that area. So those are very interesting, studies for the traffic jam in the simulation, in the computation. And there are a lot of interesting open problems for this area. It's still, in the early stage. And you know, there are many other types of the traffic jams. As we mentioned earlier, there is another category of the traffic jam introduced by motors. So this could be modeled in our system by changing, you know, setting proper, you know, attachment rate and detachment rate. So that could be a direction for our future work. In addition to this, we are talking to the clinical people in Mayo Clinical, Florida. And then we wanted to, they have a very nice study of the tau protein related to Alzheimer's disease. They have a long-term, you know, study, you know, for many years over there. We are talking to each other, trying to see how can we validate with each other, you know, between the computation and also experiments to try to understand the neuron degeneration disease, you know, certain features of the tau protein in a better way, in a systematical way. We have several papers published, you know, along this direction, and also we published our source code and the data in the GitHub link. If you are interested, you are welcome to take a look at. So finally, I want to bring your attention that we are managing a journal, Engineering with Computers. So we welcome everyone to submit your future paper writings to us. And if you are interested in organizing a special issue on related topics within the scope of the journal, you're welcome to email me. Thanks for your kind attention, and I'm very happy to take questions.